Hello, aye. The anatomy of the pupillary dark reflex. Dark reflex? It's not something people talk about. It's also not something that's been studied brilliantly. But it is the opposite of the pupillary light reflex. It is sympathetic nervous system mediated. It is not only interesting anatomy, but clinically relevant because it explains how issues in the chest cause signs up in the eye. But yeah, this is what we're going to look at. Oh, I've been talking and teaching all day and my voice is tired and sore, my head is tired and sore. Let's see how I get on with this. It's an interesting topic, mind. So the eye has a number of reflexes. The eye is a very busy place anatomically and neurologically. And in terms of the pupil, the eye is adapting the diameter of the pupil to manage the amount of light falling on the retina inside the eye, right? And we saw that with the pupillary light reflex, with a bright light, the pupils constrict and less light enters the eye. And the pupillary dark response or dark reflex is the opposite of that then. When you move into a dark space, the pupil dilates to let more light in. This isn't the same as dark adaptation. So if you spend a long time, minutes in a dark space, the cells in the retina will adapt. Your eyesight will improve slowly over tens of minutes in the dark, so you'll be able to see better. If you go stargazing, you'll know you have to wait for quite a long time for that to happen. But in terms of the pupillary dark reflex, well, we've got the iris, we've got the pupil, which is just a hole. It's black because it's dark in here. The light goes in and light falls on the retina back here. The retina is made up of rods and cones, which sense light and give you vision. And the rods and cones pass that information to another layer, the retinal ganglion layer. And in the retinal ganglion layer, we also find another population of cells, the intrinsically photosensitive retinal ganglion cells, which can themselves respond to the intensity of light. When we talk about a reflex, we talk about the sensory bit and then the sensory limb, the afferent limb, and then we talk about where the reflex happens. With these sorts of things, we're talking about the brain stem, a little bit of hypothalamus today as well, and then we talk about the motor limb of the reflex, the efferent part, which today is going to be the sympathetic nervous system. If you know a bit about the anatomy of the sympathetic nervous system, you know that this isn't going to be a simple brainstem and back reflex. So in terms of the sensory limb then, light falls onto the retina, and it's probably the rods that are involved in this pupillary dark response, and certainly the intrinsically photosensitive retinal ganglion cells, which are responding to the intensity of light entering the eye and sending that information through the optic nerve. So the optic nerve, cranial nerve 2, is the afferent limb. So the optic nerve has its own canal. So the optic nerve will pass through the optic canal to get into the cranial cavity, and it's up against a whole bunch of really interesting and important structures here. It doesn't have to travel very far. Here's a nice colourful brain. So cerebral cortex, here's the brain stem down here, pons, medulla, and we've got a lovely colourful optic nerve entering from either side here. And the two optic nerves come together at the optic chiasm. And some of the fibres cross to the other side of the brain and some of the fibres stay on this side of the brain. Nasal cross and temporal stay on this side. And then that continues as those fibres then continue as the optic tract, bundles of fibres in the central nervous system, we like to call them tracts. And there's the medulla oblongata, there's the pons, there's the midbrain, there are the superior and inferior colliculi, which the superior colliculus is involved in vision. That visual tract is running around the midbrain. So it's gonna contribute fibers to midbrain structures as well as to the visual cortex. And actually, if that's the chiasm there, if that's the chiasm there, that's the pituitary gland, that's the hypothalamus there, and that's the thalamus. And in the hypothalamus, just superior to the optic chiasm, there is a nucleus, or maybe a group of nuclei, called the suprachiasmatic nuclei. Now, we don't entirely know how this reflex works, but we do know that light intensity from the eye passes to the suprachiasmatic nucleus and is involved in circadian rhythms. That is the rhythm of day and night and our response to light and dark through the day, which makes us go to sleep and wake up and has a whole bunch of other effects on the body. So the intensity of light passes to the suprachiasmatic nucleus in the hypothalamus and passes to other regions of the brain that are involved in circadian rhythms. Do those also relay to other centers of this pupillary dark reflex? Maybe, maybe not. In the midbrain, so this bit here, in blue here we can see the ventricular system, or part of it, so the third ventricle and the cerebral aqueduct flowing down here. The midbrain then, the tectum, is this region of the midbrain dorsal to the cerebral aqueduct. We see the colliculi here, those little bumps. I talked about these in the pupillary light reflex video. And there are some pretectal nuclei. And it might be that this, well, we know that this light information goes to some pretectal nuclei. It might be that this light information goes to pretectal nuclei in here, we don't know which ones, which then project out to sympathetic premotor nuclei. That is, neurons that are going to switch on parts of the sympathetic nervous system. So this is a bit woolly, so I should gloss over it really, but that light is sensed by the cells in the eye, sent to the optic nerve, and they either get to the suprachiasmatic nucleus in the hypothalamus or pretectal nuclei. Now, from those inputs, in the midbrain, there are some very nearby centers. So if the grey matter around the cerebral aqueduct is called the periaqueductal grey, there are some relays in there. Also in the midbrain, there is a region called the locus ceruleus, the blue spot. There are a lot of lovely colourful spots in the midbrain. The locus ceruleus, so it's blue because in fresh brain sections, those cells hold a noradrenergic neuromelanin, which has got a bit of a blue tinge to it. But that is a sympathetic premotor output. And then there are periventricular nuclei as well. But it seems likely that somewhere nearby in the midbrain, the sympathetic nervous system is triggered, or parts of it. Ooh, now we're on home ground. Now we're on the motor part of the limb. This is much more comfortable for me. This is some major gross anatomy. Right. The sympathetic neurons that are triggered, wherever they start, they send axons down the spinal cord to the chest. This is a model of the posterior thoracic wall, posterior neck, bit of brain up there, and so on. So this is the spinal cord running down here. This motor pathway is going to be a 3-neuron chain. The first neuron is running down the spinal cord to the T1, T2, or T3 levels. And then in the lateral horn of the spinal cord, that's going to synapse with preganglionic sympathetic neurons, which will leave the ventral horn of the spinal cord. And out here, this is the sympathetic trunk. These are the sympathetic ganglia. So that second neuron will run from the spinal cord out to the sympathetic chain, and it'll send up to the superior cervical ganglion. And in there it will meet the third neuron, the postganglionic sympathetic neuron, which will, oh, those are the arteries there, which will, so the sympathetic neurons, they run throughout the body and they often follow arteries as much as they run with nerves. And what we'll see is that these sympathetic neurons get into the head by following the common carotid and the internal carotid arteries. So there's the common carotid artery. So sympathetic, when we look at these major blood vessels, we find them covered in like a fine filigree of neurons. And we'll see sympathetic neurons running with the internal carotid artery. The internal carotid artery goes into the skull through the carotid canal, and then it runs anteriorly, and it puts the internal carotid artery just posterior to the orbit. So those sympathetic neurons, that third neuron in the chain of three, is brought just posterior to the eye, at which point these neurons jump off and they travel with the ophthalmic branch of the trigeminal nerve. They're just all going into the orbit, so they just travel with another nerve that's going in the same direction, right? So the trigeminal nerve is the sensory nerve of the face, and the ophthalmic branch is going up here into the eye and what have you. So these sympathetic neurons just get carried along through the superior orbital fissure into the orbit. Big eye model. We're almost back at the eye now. That is the internal carotid artery, and those are the sympathetic nerves covering it. There goes the ophthalmic branch of the trigeminal nerve over here. Do you remember when we looked at the pupillary dark reflex, we saw the ciliary ganglion, which is a ganglion, it's a parasympathetic ganglion. No use for that today. We don't need that. We've done all our ganglioning, thank you very much. What we have is we have the long ciliary nerve here. So the long ciliary nerve is, the ophthalmic branch of the trigeminal nerve is going to send sensory fibers to the anterior eye. That's how the anterior eye is so sensitive. So the long ciliary nerve is going to carry those fibers, but the sympathetic neurons, those postganglionic sympathetic neurons, are also going to run in the long ciliary nerve to get into the eye, and then they'll run around the eye, and then they're going to run to the dilator pupillae muscle. That's the trigeminal nerve, cranial nerve 5, V1, V2, V3. So that's the ophthalmic branch running up there. So those branches I was pointing out in there are branches of the branches. But that's how it often gets explained, like the sympathetic nerves are part of the ophthalmic branch of the trigeminal nerve, which can be very confusing. But that's all it means, is those sympathetic nerves are coming up from the chest, and they're using these nerves to get to the eye, because they're all going to roughly the same place, all right? Oh, long ciliary nerves into the eye, dilator pupillae. Right. So we're now back in the eye, and we're on the deep surface of the iris, and the dilator pupillae muscle, it's not actually the rays we're looking at there, but it's like that. That's actually going to the lens. But on the deep surface of the iris, there are muscle fibers running radially. So when they contract, they will pull the iris more widely open. So this is an active thing. Smooth muscle fibers are triggered by those sympathetic nerves to contract, to get shorter, and in doing so, the iris opens widely. So in terms of the pupillary dark reflex, a reduced amount of light falls on the rods and the cones and the intrinsically photosensitive retinal ganglion cells. That information is travelled through the optic nerve. Some of it crosses over, some of it doesn't at the optic chiasm, passes to pretectal nuclei, the suprachiasmatic nucleus maybe in the hypothalamus. Some relays somewhere trigger sympathetic outflow, probably from the midbrain, maybe from the hypothalamus, down the spinal cord to those thoracic levels, which trigger the sympathetic neurons, which run up to the superior cervical ganglion, which then trigger the postganglionic sympathetic neurons that then run all the way to, it's a long way, isn't it? All the way to the dilator pupillae muscle and cause your pupils to dilate. Now, being part of the sympathetic nervous system, this means that your pupils can dilate for other reasons than just sitting in a dimly lit room. So parasympathetic innervation causes the pupil to constrict, sympathetic innervation causes the pupil to dilate, which means that there is also an argument that maybe pupil dilation just occurs if you turn down your parasympathetic innervation, because these two things are working in opposition. Who knows? But certainly your pupils can dilate as part of the fight or flight response. If you let more light onto the retina, the rods and cones, those rods that are very sensitive to low light conditions, when you see in the dark, it's the rods that are doing most of the work. When you let more light into the eye, you have greater sensitivity. You might spot something you hadn't spotted before. So there's a threat, sympathetic nervous system, adrenaline gets dumped and everything gets switched on, your pupils dilate. That's what triggered my fear response, that terrifying thing over there in the dark, and I'm going to run away, which is cool. But also, your pupils can dilate with mental effort, like you're working on something, your pupils dilate. Your pupils can dilate as part of like an arousal mechanism. The more excited and aroused you are, the more your pupils can dilate. And there are other stimuli as well which can cause dilation of the pupils. Your pupils can dilate in response to sound. So clearly there's some complex wiring. There are some more circuits in there controlling all of this that we don't know about. This is just not something that's been studied fully. Hey, if you're interested in if you're into circadian rhythm research, maybe you should expand that into looking at the pupillary dark response. Anyway. Also, drugs can dilate the pupil. This can be really useful because if you want to examine the retina, you can dilate the pupil and get a better view. So dilating the pupil, dilated pupils is also called myodriasis, by the way. But anticholinergics, so that switches off the parasympathetic innervation, or adrenergic agonists, which will switch on the sympathetic synapse receptors. Those can both cause dilation of the pupils. Stimulants can cause dilation of the pupils. But one of the reasons all of that anatomy that I described is so important is because if somebody has anisocoria, or if somebody has a dilated pupil just on one side, anisocoria is when the pupils are different sizes. If the pupil is dilated on just one side, then is that sympathetic reflex not functioning normally? What I'm talking about here is Horner's syndrome. You notice that the eyelid is drooping on one side, ptosis. The pupil is constricted, miosis, and maybe there's a loss of sweating on that side of the face, anhidrosis. Those are the three things that lead you towards Horner's syndrome. Those three things all suggest that the sympathetic nervous system on that side isn't working. So sweating, sympathetic nerves innervate the sweat glands, tell them to do their thing. There's a muscle up in the eyelid that's innervated by sympathetic nerves, which helps keep the eye open. So if that has lost its sympathetic innervation, the eyelid droops a little bit, ptosis. And then if pupillary dilation is sympathetically mediated and pupillary constriction is parasympathetically mediated, if you've lost the sympathetic side of that, then the parasympathetic innervation is going to cause the pupil to constrict. This might give anisocoria, different sized pupils on either side. That's the anatomy that explains that. Now we're worried because we've seen this in the eye, that there's something wrong in the chest. Is there a tumour pressing on part of the sympathetic nervous system, on those sympathetic nerves that are running to the face and the eye? Is there some pathology in one of the carotid vessels which is compressing those nerves against another structure and stopping them working properly? What's causing this? Maybe it's something that's, fairly benign, but maybe not. So the eye can be a window into pathology in the chest because of that anatomy. But it's just downright interesting. Why does a reflex detecting light from the eye go all the way down here and back up again to do the thing? Well, there you go, that's evolution for you. But I hope that was interesting. The eye is a fascinating bit of anatomy, and I'm sure we've got plenty more reflexes to talk about in the future. See you next time.